Hello, this is cellular respiration by Mark England. Cellular respiration has four main steps: glycolysis, which is the first, pyruvic oxidation, which is the second, citric acid cycle, also known as Krebs cycle, is third, and oxidative phosphorylation is the fourth. So, glycolysis has 10 major steps, and since it is the first step of cellular respiration, it does not make much ATP. So the first step in glycolysis is when the cell takes glucose from the cell cytoplasm. It adds a phosphate from ATP, and it yields ADP and glucose 6-phosphate. In step 2, an enzyme converts the glucose 6-phosphate into an isomer, fructose 6-phosphate. So pretty much, it's just the same thing as the first step with a different enzyme. Step 3, however, is a continuation of step 2, and where a different enzyme uses another ATP molecule to transfer the phosphate group from fructose 6-phosphate to form a fructose 1,6-biphosphate. It's kind of similar, but they're also very different. So, in this diagram, you can see that it can form either GADP or DHAP, and I'll explain that in a little bit. Another different enzyme splits the fructose 1-biphosphate into two different sugar isomers. Alright, so these isomers are very similar, but the definition of an isomer is that they have the same molecular formula, but they are just arranged differently, so they look similar, but they actually are very different. So step 5 has a molecule that changes these two phosphates into a different molecule. The name's really hard to pronounce, so I'm just going to kind of skip it, but just keep that in mind. In step 6, an enzyme transfers a hydrogen over to become the oxidizing agent in NAD+ in order to form NADH. Next, it adds a phosphate from the cytosol to the oxidized new phosphate, which is really hard to say. And this occurs because it was made from step 5, so it just builds on after that. So step 7, it just takes a P from the new molecule that was just made, and it changes from ADP to ATP. And since there's 2 of these new molecules, there's 2 new ATP molecules. Step 8 is just a rearrangement of the carbon atoms in this molecule. So it just changes over to that. It's really hard to pronounce, so I'm not even going to try. And then in step 9, water is released to form PEP. In step 10, the PEP transfers a P to ADP to form pyruvate and an additional 2 ATP. So in summary, the net total of what was produced in glycolysis is 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of water. Up next is pyruvate oxidation. So pretty much, there's 3 small steps in pyruvate oxidation. The pyruvate that was made in glycolysis is now sent through the mitochondrion, and a carboxyl group is removed from the pyruvate, releasing CO2. Then an NAD+ is reduced to NADH. Finally, an acetyl group is transferred to coenzyme A, resulting in acetyl CoA. Pretty much, this is a setup for the Krebs cycle because no ATP is produced in this step. So, in a nutshell, the Krebs cycle is used to get rid of CO2, create some ATP, and collect some electrons for the ETC. It does this by getting the acetyl CoA from the pyruvate oxidation, goes through a few steps, loses the CO2, but creates an NADH, loses some more CO2, but creates some more NADH. And as it goes on through the cycle, it creates some ATP, some FADH2, and some more NADH. Now it is time for oxidative phosphorylation. This is the biggest step in cellular respiration because it contains the electron transport chain and part of chemoosmosis, which is a combination of these two diagrams. Electron transport chain uses the electrons that were gathered from the Krebs cycle, puts them into this membrane, and then at the end result is water is created from the intermitochondrial membrane. While all of this is going on, some ATP has been made from the phosphorus that was donated during the earlier cycles. So therefore, this is one of the most effective way to make ATP because the ATP synthase is here and it makes some more ATP. And then with all the electrons donated to create water, it is just the most efficient way to get energy in the cell. So, a summary of the cellular respiration is that glycolysis occurs in the cytosol and it breaks glucose into two pyruvates. Then it goes to the pyruvates oxidation, which is pretty much a setup stage for the Krebs cycle. The Krebs cycle takes place within the mitochondrial matrix and breaks down the pyruvates into CO2 to produce some ATP and NADH. So, between glycolysis and the Krebs cycle, some reduction occurs and it reduces NAD+ into NADH. That's how you get that NADH. This is a byproduct of the glycolysis and it is then transformed into that. There is very little ATP produced in these steps, but it won't matter because the electron transport chain is what produces most of the ATP. So the electron transport chain accepts all of the electrons from NADH and passes them from one protein molecule to another. At the end of the chain, the electron combine with H+ and O2 to form water, and this releases energy. This is what creates the ATP. This energy is used by the mitochondria to synthesize 90% of the cellular ATP via ATP synthase, through a process called oxidative phosphorylation in the inner membrane of the mitochondria. So, using ATP synthase, it's able to fully utilize what is made of the ATP, and that is called the oxidative phosphorylation. The end.